For my presentation today, I'll be exploring how different evolutionary forces contribute to the evolution of venom in the red diamond rattlesnake. I want to begin by discussing coevolution, as coevolution is often viewed as the predominant driver of adaptive trait variation between interacting species. So here we see what is often used as a textbook example of a predator-prey coevolutionary dynamic. These rough-skinned newts near the west coast of North America produce tetrodotoxin, or TTX, which can be lethal to predators. And then this TTX production varies spatially across its range. Garter snakes that prey on the newts have in turn evolved a resistance to TTX, which also varies spatially. Comparing the spatial variation between the two traits, we see that toxicity and resistance vary concordantly with one another between these two species. So it was previously thought that variation in TTX production and TTX resistance was likely co-evolving due to an evolutionary arms race between predator and prey. Recent research has found that while toxin resistance in the snake is higher than what would be expected under neutrality and population structure, as we can see in this plot, where the red line indicates TTX resistance and the gray dotted line represents neutral evolutionary conditions. Newt toxin production varies geographically with what would be expected under neutral conditions, as indicated here. Therefore, while TTX resistance in the snakes is clearly the result of selection, non-adaptive forces play a major role in explaining newt toxicity. So although we may think selection as a result of coevolution is producing variation, neutral processes and underlying demographic processes may be just as important in driving variation. And as I mentioned, this is often the textbook example of a predator-prey coevolutionary dynamic. However, when we actually test for the role of non-adaptive contributions of trait variation, we find that it plays a major role in this system. So what about other systems where we assume coevolution is occurring? Snake venom is another example where coevolution is often assumed to be occurring. So snake venom often exhibits this extensive variation, both interspecifically and intraspecifically, such as this example of the South Pacific rattlesnake. So these HPLC chromatograms represent the venom composition of a rattlesnake from different populations, where each peak corresponds to a unique toxin family labeled below. As we can see, venom varies between populations in this species, which begs the question, why would this variation occur? Now, it is often assumed to be the result of coevolutionary interactions with prey, as venom plays a direct role in subduing and consuming prey. However, the role of other evolutionary mechanisms, especially that of neutral processes, are often ignored. In general, snake venoms have emerged as an ideal system for studying adaptive evolution because it exhibits variation at multiple levels, as we can see here. It also plays a direct role in the fitness of the animal. As I mentioned, venom plays a specific role in obtaining food for the snake. And it's a complex polygenic trait, and this is important as most ecologically important traits are likely polygenic. And lastly, it's genetically tractable, so genes responsible for venom composition are only expressed in the venom glands. As previously mentioned, snake venom variation is frequently assumed to be the result of coevolution, but this is often not tested. So using snake venom, I want to investigate what evolutionary forces drive variation of this trait or any complex trait relevant to fitness. Now, because snake venoms do play a direct role in obtaining prey, I predict that venom evolution will be driven by selection due to interactions with prey. However, I also predict that neutral evolutionary processes, such as isolation by distance and genetic drift, will also play a major role in the variation of this trait. This is because genetic drift alters the spatial distribution of alleles, which can potentially interfere with local adaptation. So in other words, I predict that complex traits evolve due to a complexity of evolutionary forces. The species I'll be using to investigate this question is Crotalus ruber, or the red diamond rattlesnake. So Crotalus ruber is endemic to Baja California, Mexico. It's found throughout the entirety of the peninsula and reaches as far north as Los Angeles, California in the United States. It is divided into two subspecies, one in the north and one in the south is indicated here. And it's overall a large rattlesnake which feeds primarily on mammals, occasional lizards, and rarely birds. The Baja California Peninsula itself is a fascinating area of ecological diversity due to its unique climate and terrain. It is a desert environment with high annual mean temperatures and low precipitation, but it features this unique inverse wet-dry season from the northern half of the peninsula to the southern half. So northern Baja receives the majority of its rain from winter storms from the West Pacific, whereas the southern Baja receives its rain from these southern summer storms, and it receives a greater amount of rain than the north. So this unique abiotic variation would potentially affect species distributions or population structure of a single species throughout the peninsula. So we sampled Crotalus ruber throughout its range. We sampled 16 individuals, and we extracted blood for DNA, venom, and venom glands for transcriptome analyses, RNA-seq. And we measured each snake from the snout vent length. We also downloaded additional genomic data from previous studies. We started by generating a reference genome of Crotalus ruber using one of the individuals we sampled. So the reference genome would allow us to identify venom genes, map genomic and transcriptomic data of other individuals more accurately, and quantify the expression of venom genes in any individual. So we assembled a highly contiguous genome, and here we see a chromosomal representation of the genome featuring 7 macrochromosomes, a sex chromosome, and 10 microchromosomes. This outside shows toxin gene families and their position on the genome. We then combined SNP data from 7 whole genome samples with the SNP data for 34DD RADseq data from a previous study, and used these data to estimate population structure. We used the program construct and found that k equals 2 was the best fit for the data, with populations separated between the north and the south of the range. And this separation is actually consistent with subspecies delineation. We then used these data to calculate FST and obtained a value of 0.295, indicating substantial genetic differentiation between the two populations. We then sequenced the venom gland transcriptomes for 13 individuals and estimated expression of the venom genes using transcript per million, or TPM. So we ran a PCA on these data, and we can see that individuals separate on PC1, which has 43% of the variance, primarily due to size. So individuals on the right here are 65 centimeters or below, and individuals on the left are larger than 65 centimeters. PC2 appears to capture some geographic variation as well. So to further visualize venom composition across our different samples, we grouped the venom genes by families, and the proportion of total toxin family expression was calculated and represented as a pie chart and placed at the location where sampled. And SVL is included under each chart. And here we observe variation geographically and ontogenetically. To visualize the ontogenetic change, we can look at a single population here in Loreto. We see that smaller individuals' venom composition are dominated by myotoxin, labeled here in red. The larger individual's venom composition is dominated primarily by SVMP in blue, CTL in the teal, and PLA2s in pink. Geographically, we see an adult individual here in the north whose venom is dominated primarily by the SVMP, CTL, and PLA2. In the south, adult individuals appear to have a greater proportion of myotoxins in their venom. So to determine whether venom expression variation was significant geographically and ontogenetically, we ran the program DE-seq2. So starting with ontogeny after accounting for geography, we see 24 venom genes that are significantly differentially expressed between adults and juveniles. We use 65 centimeters as that cutoff. And we'll see many of those genes belong to the toxin families we saw in the previous slides, such as SVMP, CTL, and PLA2. Moving to geography, after accounting for size, we see 5 venom genes that are significantly differentially expressed, also belonging to those toxin families previously identified, SVMP and CTL. So we've estimated population structure using neutral genetic differentiation, and we've explored venom expression variation. But now we want to determine what is actually driving venom expression variation in this system. The underlying assumption would be that this variation is likely driven by selection. So we conducted a conditional redundancy analysis using venom expression PC1 from our initial PCA as our response variable. The explanatory variables are neutral sequence variation, toxin sequence variation, prey availability at each site based on diet studies for the species, environmental conditions at each sample site based on world-clim bioclim variables, and size of the snake. The following chart shows the results of the RDA, starting with the marginal model at the bottom, which represents the best model for our data. In this case, both SVL, or size of the snake, and neutral sequence variation were the best factors for predicting most venom expression variation. SVL, when conditioned for all explanatory variables, was significant, and neutral and toxin sequence variation were also significant. So from this, we have found that Crotalus ruber has two genetically distinct populations, one in the north and one in the south. Crotalus ruber also undergoes significant ontogenetic and geographic venom variation. And most importantly, life history or selection across time, along with neutral evolutionary processes, explain the most variation in venom expression. And these factors were more important than geographically variable abiotic or biotic factors. So from our study, we can conclude that variation in extended phenotypes is not always the product of coevolution. So as we saw in the newt garter snake system, and as we've seen here, neutral evolutionary processes can play just as an important role, if not more important, in shaping the variation of extended phenotypes. We also found that neutral evolutionary processes and selection can both play a role in determining variation of complex traits. So any study looking at factors that may impact the variation of traits should always consider the role of multiple evolutionary processes and selective pressures. Evolution of extended phenotypes is not likely driven by a single coevolutionary dynamic. So what's next? Well, there are 3 other species of rattlesnake found throughout the region, so we plan to investigate whether the same factors or different factors shape venom evolution in all 4 species in similar or different patterns. We also investigate how competition between these species may impact venom variation. I'd like to acknowledge all these awesome individuals for their contributions to this project, as well as the agencies and institutions for helping fund this research, especially the National Geographic Society. Thanks so much for attending my presentation, and I'd be happy to answer any questions you might have.